Right. Hello, everyone. I've decided to record my talk to lower the risk of any glitches from this marvelous technology. By the time you hear me, it will be close to midnight in South Africa, but it's much nicer to give a talk at midday in my garden. Right, first let me mention the most important acknowledgements. Danny and Gary, thank you so much for allowing me to participate in this regional meeting despite my geographical location. It's a great privilege to be part of this meeting, and this is definitely one of the great advantages of a virtual meeting. I'd also like to acknowledge Michał Gwizdała up front because he was the mastermind behind most of what I'm going to present. Today I'll be speaking about phycobilisome, which is the main light harvesting antenna of cyanobacteria and red algae, and has been studied for the past 55 years. So shouldn't we know everything about this complex by now? Well, there are quite a few important details that have remained hidden until we started to investigate individual complexes. In this talk, I'll focus on phycobilisome from the cyanobacterium Synechocystis PCC 6803. Here you can see a toy model of this complex, showing a few core units and 6 protruding rods. This huge complex binds almost 400 phycocyanobacterium pigments whose spectral properties are drastically tuned by the protein environment, covering a broad range of absorption and emission energies from the rods, called phycocyanin, to the core, called allophycocyanin, to the terminal emitter subunits in the core. After light absorption, there are three competing fates of the excitation: fluorescence, heat dissipation, and transfer of the excitation energy to the reaction center via the photosystems. Now, when we isolate this complex, fluorescence and heat dissipation remain as the only two de-excitation channels. So when there is a quencher somewhere in the complex, a state that quenches an excitation non-radiatively, the amount of heat dissipation will increase and the fluorescence and intensity and lifetime will decrease. In other words, there's an inverse relationship between fluorescence and the amount of heat dissipation, a relationship that has been widely used for decades, considering fluorescence as a probe of photoprotection. Now, one challenge when you do an ensemble experiment is that you measure the average spectroscopic properties of perhaps a few million or billion complexes in all sorts of conformations associated with varying degrees of quenching and spectral properties. For example, a bulk fluorescent spectrum of a light harvesting complex at room temperature is typically broad and featureless, but a lot of information may be hidden below this broad peak. So in these ensemble measurements, it becomes impossible to disentangle the individual conformational and spectral states and the associated dynamics. And that's why we need a probe that allows you to monitor the state of individual proteins. And this probe is single molecule spectroscopy, a technique that allows you to investigate the complexes one at a time, which enables the ultimate level of sensitivity by revealing unsynchronized dynamics, statistically rare events, separation of static and dynamic heterogeneity, and one is able to obtain a full probability distribution of observable properties instead of just one average value. Let's see what hidden dynamics phycobilisomes display. I'm going to show three examples. The first being emission dynamics. Here on the left is an example of a fluorescence trace of one phycobilisome complex, where we have simultaneously captured the fluorescence intensity in black and the fluorescence lifetime shown in red. And we've also resolved the intensity levels shown in blue. The first interesting observation is the occurrence of abrupt changes or switches between quasi-static levels. The second observation is that basically all these intensity and lifetime switches are correlated, as shown here on the right. This plot on the right shows us two things. First, the slope tells us how large the complexes are, ensuring that we worked only with intact complexes, because they fall apart quite easily in this environment. And secondly, the linear correlation tells us that thermal energy dissipation occurs mostly at the terminal emitters, that is, the phycocyanobelins in the core that contribute most to the fluorescence emission. We studied the fluorescence intensity switches across a broad range of excitation intensities. Shown on the left is an example of a trace at low light and one at high light intensities, indicating a huge difference in the switching frequency. Let's parameterize the switches. We define an intensity decrease as a switch from an unquenched to a quenched state, and an intensity increase we define as a switch from a quenched state to an unquenched state. Now, plotting the switching frequency as a function of excitation intensity, we notice that the frequency of both switches increases, meaning they're both light-induced. It's important to note that we went down to extremely low excitation intensities corresponding to physiologically relevant light intensities, the green dotted line being at 2000 microeinsteins. It's the first time such low intensities were used in a single molecule study. Now, when you look carefully, you'll notice that the black plot is steeper than the red plot. Now, when I take the ratio between the two, this ratio also increases with enhanced illumination, indicating that the net effect is an equilibrium shift toward quench states, exactly what one expects from a photoprotective mechanism. More light means more quenching. So this is a new intrinsically available photoprotective mechanism that is switched on immediately and with greater effect as the excitation intensity increases. Now, what is the underlying molecular mechanism? First, when you increase the amount of glutaraldehyde, a chemical crosslinker, the switching frequency decreases significantly for both types of switches, indicating that the intensity switches are governed by protein conformational dynamics. Secondly, by simultaneously measuring fluorescence spectra, we observed a fascinating phenomenon. The complexes occasionally displayed enhanced red emission, and in 80% of the cases, this red emission was preceded by a long-living quench state, which means that the quench state must be associated with the red emission state. This correlation required further investigation. We used stark fluorescence spectroscopy, and by this we managed to resolve a far red emission state that has a strong charge transfer character. So if this quench state, which was shown in the previous slides, if it's related to this red state that's resolved by stark spectroscopy, it must also involve a charge transfer state. Let's now consider a simple model containing three compartments, one for the rod pigments, PC, one for the core pigments, APC, and one for a single pigment in the core. When this pigment in the core, when its charge transfer state is accessed, the excitation energy is quenched. But when a protein conformational change allows the charge transfer state to mix into the pigment's S1 state, red emission appears. On the right is a slightly expanded version of this model, where I've separated the 660 and 680 nanometer emitting pigment pools in the core. And doing this, I managed to reproduce the experimental data pretty well. In this plot is shown the degree of quenching as a function of the peak position of the red emission. Finally, it's very important to note that the red states aren't artifacts due to the experimental environment, because the average of all single molecule spectra, the black line, black spectrum, perfectly matches the ensemble spectrum shown in blue. While there's a clear difference between the bulk spectrum and the single molecule average spectrum devoid of red emission bands, indicating that the red emission must form part of the bulk spectrum. So to summarize this first part of the talk, we have been able to resolve a new way, possibly used by nature, to control light harvesting. The principle is quite simple. A change in the light intensity shifts the equilibrium between light harvesting and thermal energy dissipative states. The site of the quencher is mostly in the core, but in principle, any subunit can be the site of quenching. The molecular mechanism is summarized as light induces protein conformational change, which in turn gives rise to a configurational change of only one of the embedded pigment molecules, the core pigments, which allows a charged transfer state to mix with excited state, leading to energy quenching. Okay, in the second example, we'll look at the main mechanism of non-photochemical quenching involving the orange carotenoid protein, also called OCP. OCP is first activated by blue light from an inactive orange form to a photoactivated red form. On the right are the spectra of the two forms. After and only after photoactivation, OCP binds to the core of the phycobilisome complex where it quenches the excitation energy. Here on the right you can see the spectra of the unquenched light harvesting form of phycobilisome in red and the quenched states in blue, and these are the bulk spectra. Let's now look at the binding of a single OCP to a single phycobilisome. When using wild-type OCP, we can see in real time that upon blue light illumination, how the fluorescence is switched off. This is definitely an OCP-induced quench state because the lifetime of this quench state is significantly shorter than that of the quench state shown in the first part of the talk, and the time spent in the quench state is a few orders of magnitude longer. Now, when using an OCP mutant with a low binding affinity, we see not only how the fluorescence is switched off, but also the full recovery of the fluorescence in real time. It's really fascinating that this can be observed in real time at the single protein level. And what's even more fascinating is the fact that we can see short-living intermediate states between the unquenched and the quenched states, both when the fluorescence switches off and when it recovers. The spectrum of this intermediate state, shown in left in blue, contains two bands, one very blue shifted band and one very red shifted band, signifying again the presence of charged transfer states. On the right is plotted the amount of quenching versus the size of the blue shift of this blue band here on the left, where the lines in this plot are different models for the intermediate state compared to these two experimental data points, which correspond to the average properties of the wild-type OCP phycobilisome complex and the mutant, respectively. These experimental points match two of the model calculations. The two gray dashed lines depict the case when all the rods are 10 times, 20 times more loosely coupled to the core, and the blue line depicts the case when two rods are temporarily unbound. In contrast, the black line in this plot is the case when all rods are tightly bound to the core, and these black experimental data points, they are those of the fully quenched states. The position of these data points of the intermediate state signifies a docking mechanism of OCP upon binding and unbinding from the phycobilisome core. How the docking mechanism works, according to our data, is that after blue light activation of OCP, when it encounters a phycobilisome complex, at least one of the phycobilisome rods temporarily weakens its coupling to the core, possibly by rotating or physically moving slightly away from the core to create space for OCP binding. Now, in the third and final example in this talk, we'll look at isolated phycobilisome rods, the most blue-shifted unit of phycobilisome, emitting mostly at 650 nanometers at room temperature. When you illuminate a single rod, its fluorescent spectrum jumps between two states, with spectral peaks about 20 nanometers apart. The two states have the same average intensity, which means that both are light harvesting states. And again, the presence of glutaraldehyde decreases the switching frequency, again signifying the presence of protein conformational changes. So what's the significance of this redder state, which has not been reported before? A few years ago, it was found that in another cyanobacterium species, Anabena, a rod can bind directly to PS1 via the CPCL linker protein. Here are shown the blue and red emission spectra of the rods, which we have resolved, in comparison with the absorption spectrum of PS1 in green. Compared to the blue form, the emission spectrum of the red form has a 40% increase in overlap with the absorption spectrum of PS1. So we propose that this red form, which enables the rods to efficiently transfer energy directly to PS1, instead of first via the core complexes. And then the linker proteins serve to tune the equilibrium between the two states that are available for phycocyanin, either direct transfer via the core complex to the photosystems or direct energy transfer to PS1. So to wrap up, phycobilisomes are dynamic systems, and these dynamics are hidden from ensemble studies. In the first part of the talk, I've shown you that we have resolved a new intrinsically available light-induced quenching mechanism, which is governed by protein conformational changes. It involves a Tjaart's transfer state on a single pigment in the core, and it is present even under physiologically relevant light intensities. In the second part, I showed that OCP induces a transient intermediate state when binding to the phycobilisome core. And in the last part of the talk, I showed that the spectral flexibility of the rods allows them to play various roles in the photosynthetic apparatus. So let me acknowledge everyone who has contributed to this work. So especially Michał Gustauer, who has done the experimental work, some of the data analysis. Joshua Buerta has written some of the codes for the data analysis. Some of the measurements were done in the lab of Brink van Grondelle at the Free University in Amsterdam. The samples were isolated and provided by this group in CI Saclay in France. And the stark measurements and data analysis was done by Wahadu and his students in Bangladesh. And of course, I want to acknowledge all the funders that have contributed, without which none of this research would have been possible. And finally, our single molecule spectroscopy setup in Pretoria with the group members. And last of all, thank you very much for listening. I would be very glad to answer any questions.